Welcome to video 2 of topic 3 IB Biology Genetics. In this video we are looking at chromosomes. Before we begin, we just want to recognize that whole idea of the essential idea for this section, and that is that chromosomes carry genes in a linear sequence that is shared by members of the species. So all species have got the same number of chromosomes and the same genes on those chromosomes. We talked about genes in our previous video. Now what this is saying is that those genes are carried in this linear sequence along the chromosome, as we already know. We're going to look a bit deeper into that in this video. So here we are with this picture again that we've seen in the previous video, which is showing that hierarchy from the DNA, or from the bases that make up our DNA, to the genes, which are sections of that DNA along what we call the chromosome. The DNA is wound around what we call those histone proteins to make that chromosome shape. Those chromosomes are found in the nucleus of cells. This is showing you a eukaryotic cell. Okay, just bearing in mind that in our prokaryotic cell we have a slightly different nature with how our DNA is. It's not found in that nuclear envelope as such. However, it is still made out of DNA and it is still our genetic material. So our understandings. First of all, prokaryotes have one chromosome consisting of a circular DNA molecule. Okay, so that's your first understanding, is these prokaryotes or prokaryotic cells are different to our eukaryotic cells, and the prokaryotic cell has this one chromosome consisting of a circular piece of DNA molecule. Now, this DNA is not associated with proteins and is described as what we call naked DNA. Okay, so it floats freely in the cytoplasm as such. There is only one copy of each gene because there's actually only one chromosome in our prokaryotic cells. Two copies are present very briefly just before cell division via binary fission. And remembering, binary fission is the method that our prokaryotic cells divide. Now, some prokaryotic cells also have what we call plasmids. Now, these are features that are not found at all in eukaryotic cells. Our plasmids, okay, so some prokaryotics have these plasmids, but remembering the eukaryotics do not. Plasmids are small circular extra DNA molecules. They're not connected to the main bacterial DNA and are not required by the cell under normal conditions, but they may help the cell to adapt to unusual circumstances. Plasmids are not always replicated at the same time as chromosome replication, hence there may be multiple copies within a cell. Copies of plasmids can be transferred from one cell to another and even across species. This is a natural method of gene transfer. Genetics engineers have embraced this, okay, so they now use plasmids to transfer genes between species artificially. Alrighty, so our eukaryote chromosomes are linear DNA molecules, and they're associated with those histone proteins. So remember, the histone protein is what the DNA wraps itself around to form the chromosome. So DNA in eukaryotic cells is usually in the form of chromosomes. We've spoken about this in the previous video. You would have studied this in previous years. This genetic material allows organisms to survive. It's found in unicellular and multicellular organisms. None of this is new for you yet. allows certain traits to be passed on to next generations, remembering our genes and alleles from the previous video. When the cell is not dividing, the chromosomes are not visible. It is in the form of what we call chromatin. Chromatin equals strands of DNA and histone proteins. Okay, so we've got our chromosomes here, that chromatin. Okay, we're then wrapping around, so let's go backwards. We've got our DNA bases and the base pairing that we've learned about. So then we've got that DNA double helix that wraps itself around the histones, okay, to create that chromatin that then condenses to form our chromosomes. So to compare our prokaryote and our eukaryote chromosomes. So in our prokaryote, generally there's only the one chromosome. Eukaryote, we've got two or more. In terms of shape, in our prokaryote, it's circular. Eukaryote, our chromosomes are linear. Histones are not present in prokaryotes in all cases. Eukaryotes are present. Presence of plasmids, sometimes present in prokaryotes, but they're never present in eukaryotes. Are they organised in pairs? Nope. In prokaryotes, there's not, because there's only the one chromosome. In eukaryotes, yes, they are. Remembering, if one of your standards is to know a comparison, you may be asked to compare and contrast prokaryotic and eukaryotic chromosomes. So not just saying how they're different, saying how they're different and similar and choosing different points. In a eukaryotic species, there are different chromosomes that carry different genes. Okay, so let's have a look at this whole concept of genes on the chromosome. We looked at this in the previous video, okay? So here we've got two chromosomes, one being the paternal, one being the maternal, and you can see along those chromosomes are our gene pairs, what we call alleles when they are different, okay? Knowing that two genes that are the same are called homozygous. In this case, if they're both represented as capital letters, that'd be dominant. If they're both represented as lowercase letters, that'd be recessive. A heterozygous genes alleles, okay, are when we've got two different traits. The trait is represented by different alleles, okay, a recessive and a dominant allele. So just some terminology to remember from back in year 10 there. All right, homologous chromosomes carry the same sequence of genes, but not necessarily the same alleles of all of those genes. Okay, so in a typical human cell, there are 46 chromosomes grouped into 23 homologous pairs. Homologous chromosomes refer to pairs of chromosomes that are the same size, shape, and the genes that they carry are the same. The reason there are two homologous chromosomes is that one came from the father and one came from the mother. Mother being maternal, father being paternal chromosomes. Although a pair of homologous chromosomes carries the same genes, they're not identical because the alleles, the form of the gene from each parent, may be different. The position of the chromosome on a particular gene is called the gene locus or loci, as we discussed in the previous video. Okay, now you'll hear the term one chromosome and sometimes you may get confused. Why is that? A chromosome can be double-stranded, okay? Two molecules of DNA consisting of two identical chromatids. So we're looking at A along the top there. This is where our cell is about to divide during mitosis or during the anaphase 2 of meiosis. Chromosomes can be single-stranded. This is just one molecule of DNA and is visible after our cell division. We will come back to this as we're looking at meiosis in our next video. Okay, diploid nuclei have pairs of homologous chromosomes whereas our haploid nuclei have one chromosome of each pair. So diploid 2 double, di, okay, haploid half. Let's look at that in a little bit more depth. So our diploid nucleus, which we can see on the right-hand side of the infographic there, have pairs of homologous chromosomes, 2 chromosomes of each type. They have full sets of the chromosomes for that species. Diploid nuclei in humans contain 46 chromosomes. 44 of those are what we call autosomes, and 2 are our sex chromosomes, either XX or XY. So autosomes being normal chromosomes, and then you've got your sex chromosomes. When haploid gametes fuse in sexual reproduction, the diploid number is restored. Diploid nuclei have 2 copies of every gene, apart from the genes on the sex chromosomes. Okay, so let's have a think about our haploid nuclei, and we're looking over at the chromosomes on the left of the infographic now. These have 1 chromosome of each pair, okay? They have 1 full set of the chromosomes for that species. A haploid nuclei in humans contain 23 chromosomes, 1 of each homologous pair. So 22 autosomes plus our 1 sex chromosome, the X or the Y. Gametes are what we call haploid. They need to be haploid because when the gametes fuse during fertilization, it forms our diploid nuclei. Gametes are sex cells and therefore fuse together during sexual reproduction to create a diploid cell, which we call our zygote. I just said that. Now, the number of chromosomes is a characteristic feature of a mammal of a species. So let's have a think about this concept of chromosome numbers. Okay, the chromosome number is characteristic of the species. Generally, species with different numbers of chromosomes are unlikely to be able to interbreed. The number of chromosomes may change during the evolution of a species. It can decrease if chromosomes fuse together or increase if chromosomes split. Chromosomes may duplicate, causing the chromosome number to double. However, chromosome numbers tend to remain unchanged over millions of years. And here we have some different chromosome numbers of different animals. We are looking at their diploid number, okay, so that 2N. So we've got our human at 46 chromosomes, the monkey at 42, we've got the dog at 78, the cat at 38, the mouse at 40, the frog at 26, the fruit fly at 8, the flatworm at 16, we've got corn at 20, the potato at 48, green algae at 20, we've got a fungi there with its diploid number, which is yeast at 32, and we've got mold there. Now mold has its haploid number, okay, because it is a fungi, a haploid fungi with 4 chromosomes. Okay, a karyogram shows the chromosomes of an organism in homologous pairs, showing it in decreasing length. So our karyogram shows the chromosomes of an organism in decreasing length. The micrograph is obtained from the cell during the metaphase of mitosis. The cell is stained to make the chromosomes show up. The chromosomes are arranged according to their size, structure, banding pattern, and position of their centromeres. The karyotype is a property of a cell. the number and type of chromosomes present in the nucleus. It's not a photograph or diagram of them. So the karyotype is the number and type of chromosomes present. The karyogram is the micrograph or the photograph of or diagram of the chromosomes. So the human karyogram here, we can see the 22 pairs, so the 22 autosomes, starting at chromosome number 1, which is the longest, down to 22, which are the shortest, and then we have the sex chromosomes, either the XY or the XX. So 22 autosomes and then the sex chromosome, organised in length. Human chromosomes have diploid number of 46, as we've already said. They include 22 autosomes and our 1 sex chromosomes. Human females have 2 XX chromosomes, whereas human males have 1 X and 1 Y chromosome. The X chromosome is much longer than the Y chromosome and carries more genes. A female can produce gametes, X, which contain the X chromosome, whereas the male can produce gametes, sperm, containing either an X or a Y. If an X is fertilized by an X sperm, a daughter will result, XX chromosome. If an X egg is fertilized by a Y sperm, then a son will result with an XY chromosome. Hence, the male sperm decides the sex of a child. So sex is determined by sex chromosomes, and autosomes are chromosomes that do not determine sex. So just remembering that terminology. Okay, so our sex chromosomes, and just looking at a basic Punnett square here for our sex determination, we've got the female gametes with the XX and the male gametes with XY. And you can see that if it's the X male gamete, the offspring will both be XXs, but if it's the Y male gamete, then it would be an XY, which would be male. And again, this is just the same picture, but in more pictorial representation. It's showing the effects of meiosis and then our gametes fusing to either give us an X or a Y, XY chromosome. Sorry, XX or XY zygote. Okay, looking now at your applications and skills for 3.2. First application, Cairns' technique for measuring the length of DNA molecules by autoradiography. So let's have a look at this technique. John Cairns used a technique called autoradiography to measure the length of DNA molecules in E. coli. Now, autoradiography, what this entails, cells were grown for 2 generations in a culture containing titrated thymidine. The base thiamine linked to the deoxyribose sugar. So this is used by E. coli to make nucleotides. And with a radioactive isotope of hydrogen being added to the titrated thymidine to act as its marker, or what we say the label. Now, the radioactively labeled DNA was produced by the replication in the E. coli cells. The cells were then placed on a dialysis membrane and lysed, releasing their DNA. A thin photographic emulsion was then applied to the surface and left in darkness for 2 months. During this time, some of the radioactive tritium decayed, emitting electrons, which reacted with the film. At each point where the titanium atom decayed, there is a dark grain on the film indicating the position of the DNA. The images produced by CANS showed that the chromosome in E. coli is a singular circuit DNA molecule with a length of around 1,100 micrometers. So this is an interpretation of what CANS saw. And he saw this replication of the DNA from the E. coli. So he started off with one original site of replication, and round it's gone, and then he's ended up with the 2 rings. Okay, your second application, a comparison of some different genome sizes. So you've got specific genomes there that you need to be able to compare. So when we're talking about genome sizes, viral genomes are by far the smallest, followed by our prokaryotics, and then obviously our eukaryotics. The genome size in eukaryotes depend on the size and number of chromosomes. It is correlated with the complexity of the organism, but not directly proportional. This may be because of the proportion of functional DNA is very variable, and also the amount of gene duplication varies. Okay, so comparing these genome sizes. So they're the organisms that you need to know. So you've got your T2 phage, you've got your E. coli, Drosophila melanogaster, Homo sapiens, and Paris japonica. All right, so we've got, I'll say what they are. You've got a virus that attacks E. coli, you've got gut bacterium, you've got fruit fly, you've got humans, and then you've got a woodland plant. And you've got the genome size of those. Now, remember they're talking about increasing complexity. But then what it was also saying was that there's still not the full understanding of that complexity around how much of the genome is not actually used for coding. So when you look at the Homo sapien compared to the woodland plant there, the genome of the woodland plant is much bigger, but how much is actually used for genetic coding as such? Okay, another application, comparison of diploid chromosome numbers of Homo sapiens, pan-troglodytes, Canis familiaris, Oryza sativa, and Parascarius equorum. Okay, so here we've got our 5 different species, which are the horse threadworm, rice, humans, chimpanzee, and dog. Much easier for me to say in the English name. And then you've got their diploid chromosome number. Okay, so what did you need to be able to do with them? You need to be able to have a comparison of them. So by looking at your table there, you can compare those diploid chromosome numbers. Okay, another application. The use of karyograms to deduce sex and diagnose Down syndrome in humans. Okay, so the human karyogram we've already talked about, and in terms of our sex chromosomes, we're looking at chromosomes 23 and the XX chromosome for the female. Both chromosomes are the same length, whereas the XY, that Y chromosome, is shorter. So the male chromosomes being the XY, female being the XX. Okay, now looking at this whole concept of Down syndrome, and you'll see in the karyogram here that chromosome number 21 has 3 chromosomes, okay, and we call this trisomy. We will look into this further, either in this video or the next one, where we look at a few conditions around this. However, how you identify Down syndrome from a karyogram is look at chromosome number 21, and if there are 3 chromosomes there, that does signify that Down syndrome is present. a skill, use of a database to be able to identify the locus of a human gene and its polypeptide product. So locus meaning the point on the chromosome where the gene is found, remembering. So we've already seen in our first video that there are these online databases that you can use, and we will be using this in class to be able to identify different genes on chromosomes. So there's a whole range of these databases available. They're also available for crops. Okay, so you could be looking at humans, you could be looking at various organisms. Okay, but you can, yeah, they are used extensively in agriculture as well for different crops. Okay, now your international mindedness. The sequencing of the rice genome involved the cooperation between biologists in 10 countries. Now, when you think of the crop of rice, it is grown in a range of tropical countries. It's grown here in Australia, it's grown extensively through Indonesia and Asia. Now, a group of countries needed to work together because we needed to look at the range of different rice species to be able to sequence their genome. This infographic just shows how since 2000 the development in understanding the genomes of different crops has worked. But you'll see with the rice, it started back in 2002 when they was looking at rice grain production, up to the late 2000s where they've started to actually look at rice grain yield, all based around that genome size. Thanks for watching this video, and I will see you in our next video where we look at meiosis.